Next speaker will be Dr. Arthur Haas. He's the Roland Colson Professor of the Department of Biochemistry at the University, at LSU, Health Science Center, still surviving. At any rate, his talk is entitled ISG15 Signaling and the Interferon Response. Okay.
What I'd like to talk about today is one of the two projects we have in our lab, and it's one that my group discovered some 20 years ago when I was actually still at the Medical College of Wisconsin. And it's one that only in the last 3 or 4 years has been a leap forward as people have discovered the importance of the ubiquitin and the ISG15 system and how it impacts infectious disease. So ISG15, the pointer. Oh, here it is. ISG15 stands for Interferon Stimulated Gene 15 kilodalton. And it is, as you'll see, the earliest gene induced in response to type 1 and type 2 interferons. Now, it turns out that most people don't know that LSU has a medical school in New Orleans because they think only of my esteemed colleague, Dr. Karam's medical school, which is our immediate neighbor. Tulane Medical School's down here. So we're the other medical school in LSU in New Orleans, and we are right there, and we extend over to the other side. Here's Charity Hospital, which we share teaching responsibilities with Tulane Medical School. And right here in a swath of about 24 blocks is where our new medical school is going to be, because as you'll see with the river and the proximity to the Superdome, and anybody who saw any news a couple of years ago, you immediately ask, well, if the Superdome flooded, what happened to your campus? And for about 4 weeks, this was the newest island in Louisiana, which is not real conducive to getting much work done. So that's why we're getting a new hospital here, because our current teaching hospital is unusable. But history aside, what I'd like to do, 4 things: give a brief overview of the Ubiquitin 26S proteasome system, because it really, the ISG15 system is derived from this, so it's helpful to discuss briefly how this system works. Then introduce the concept of this ubiquitin-like protein superfamily, of which ISG15 is a member and was the first one of these identified. Then the enzymology of the conjugation of ISG15 to cellular targets. And then bring up our newest thing, which is still unpublished data, and that's of a class of E3 ligases we call bifunctional or Janus ligases, which we believe is a model for how ISG functions within the cell. Okay, the details of the Ubiquitin 26S proteasome system are fairly well understood now. The Nobel Prize in 2004 in medicine was given for the discovery of this pathway. And it functions by my colleagues, Ernie Rose, who I was a postdoc with during this discovery, Aaron Sahanover, and Avram Hershko, where the 4 of us worked to work out the basic details of the pathway. The salient feature is the conjugation of this 9,000 molecular weight ubiquitin protein to targets within the cell via a 3-enzyme cascade to create a conjugate of ubiquitin, where the ubiquitin now is a transposable binding element which allows recognition of the substrate by the 26S proteasome, more specifically the 19S complex here on this 26S proteasome, with degradation of the target protein to small peptides, which can then be used for antigen presentation or be further degraded to free amino acids within the cytosol. The degradation signal, which formally is a chain of ubiquitin molecules linked typically through the lysine 48 of the ubiquitin, is cleaved off simultaneously with this degradation by an enzyme called DOA4. It is broken down to free ubiquitins by a different ubiquitin-specific protein called isopeptidase T, and the ubiquitin recycles. The ATP dependence for this process comes at 2 points. One, in a ATP-dependent unfolding of the target protein by the 26S proteasome during degradation. And the more important one comes with the activation of the ubiquitin C-terminus by an E1 activating enzyme to form a thioester, which then is transferred to an E2 protein, which transfers this thioester first onto it, and then onto the E3 ligase, which is a specificity determinant which recognizes the correct target protein, conjugates ubiquitin to it, and then constructs this polyubiquitin degradation signal. It's this area here that we've been most interested in over the last 20 years, and we've been approaching it as enzymologists looking at the kinetics of the conjugation reaction. Now, the reason that ubiquitin works so well is that it serves as a super form of phosphate, whereas phosphate only has a small information content in terms of hydrogen bonding and negative charge. Ubiquitin, being 76 amino acids, has a much larger surface and information content for forming different types of interactions with proteins. And this apparently was such a leap forward evolutionarily between prokaryotes and eukaryotes, of which this pathway is only found in eukaryotes, that nature did what she always does, and that is very quickly duplicate this system to do a lot of other things with. So very early on, ubiquitin evolved from a protein called MOA-D, which is involved in synthesizing this MoCo cofactor in one step of sulfur chemistry to produce ubiquitin. And ubiquitin, in turn, very quickly diverged into a whole series of ubiquitin-like proteins, some of which are shown here. We keep adding these things at about 1 a year so far. SUMO, NET8, HUB1, UFM1, and URM1, all of these express their biological function by becoming conjugated to proteins through their C-terminus. And they have very different functions that are unrelated to degradation. So these do not target for degradation, but ubiquitin does. So these modify protein-protein interactions. Nature got so good at this, she took another cofactor, which is another type of beta-Grass domain, Thys, which normally is involved in the synthesis of the sulfur chemistry here for thiamine pyrophosphate, and diverged that into a protein called GATE16, which also becomes conjugated to proteins, and it's involved in regulation of vesicular trafficking. All of these ubiquitin-like proteins within the superfamily have their own specific conjugation pathways which recognize only their ubiquitin-like protein and their specific substrates. Late in evolution, at the point of vertebrates, the system diverged one more time by a gene duplication to produce the protein we're interested in, ISG15, and diverged further into a second protein called FAT10, which are two-domain, based on sequence, ubiquitin-like proteins with a C-terminus here. They're both induced by interferon, ISG15 by type 1 and type 2 interferons, FAT10 only by type 2 gamma interferon. And they are involved in the immune response. And the emergence of these two isoforms, or paralogs, exactly coincides with the appearance in evolution of the interferon response. Now, 3 years ago now, my lab, John and Narasimhan, and a colleague at the medical college, Jungjia Kim and her group, published a structure for ISG15 showing that it is in fact two ubiquitin-like folds by superposition of the C-terminal ubiquitin-like domain and the N-terminal ubiquitin-like domain, shown in green and blue, with the fold for ubiquitin itself, which is shown in red, and you can see that the fold is almost identical. The RMS deviations are quite small among these groups. And using space filling modeling, you can determine what the thing looks like. It looks to be a rigid molecule where there's about 15% of the total surface area buried here in this hydrophobic interaction between these two domains. The interesting features of this molecule that we're still working out the details is this ridge of negative charge that runs along the molecule here, as well as the fact that the amino terminal domain has a face that's almost all hydrophobic on this, and we're not really sure what that does as yet. It's a C-terminal domain that probably, or most likely on mechanistic grounds, interacts with the enzymes involved in conjugation of ISG15 to proteins. So, what might this protein be doing in the interferon response? Well, if you consider the things that interferon does, the most obvious one is that it's involved in inhibiting viral replication, and that's what interferons were first discovered for. But they also are involved in stimulating lymphocyte differentiation and proliferation, particularly upregulation of natural killer cells. They're involved in upregulation of MHC class I and II proteins, as well as CD80, 86 in dendritic cells, who earn their living running the immune response. So this is involved both in innate immunity and in adaptive immunity. When this doesn't work to prevent viral replication, interferon also is involved in triggering apoptosis and killing the cell, and also acts as a proliferation and angiogenesis block to prevent further spread of an infection. And finally, because all of these things are deleterious to a cell, it's also, at some point, interferon has to downregulate its own response to shut the system down so that you don't kill the cell and induce apoptosis under normal conditions. The earliest hint of how ISG15 does this was the fact that it was similar to ubiquitin and contained the canonical ubiquitin-like C-terminus where it conjugates to proteins. And a student of mine, Keith Loeb, made antibodies against ISG15 and could show here in a Western blot stained with an anti-ISG15 antibody. If you do a time course for interferon response in A4549 human lung carcinoma cells, you see that within 4 hours, actually within 2 hours of induction with interferon, you can see the mRNA coming up and protein being synthesized. It's a very interesting response where the free protein becomes synthesized first. And from 18 to 72 hours, you see accumulation of this signal in higher molecular weight forms, which on this SDS gel shows that these are covalent conjugates of ISG15 to different cellular targets in the cell. So this obviously is what ISG15 is doing, is conjugating to proteins and somehow remodeling the signaling capacity of these cells. The induction of ISG15 mRNA follows almost exactly that for other early interferon response proteins such as MXA1 or MXA. And this later response we can show results from transcriptional events that occur early on, but the translation only occurs later, so that we knew at the time there were additional factors being synthesized late on in the interferon response involved in its conjugation. What we now know is that the mechanism for ISG15 conjugation is analogous to that for ubiquitin conjugation. There is an activating enzyme, E1, which Yuan and Krug has shown is an E1-like protein called UBE1L that is absent in most lung cancers, and it's present in, on chromosome 3 within a region that's deleted in a large number of lung cancers, and it's long been thought that there was actually a tumor suppressor gene within this area, and that tumor suppressor gene turns out to be the gene for UBE1L. There is an E2 protein which is going to transfer this activated ISG15 from the E1 activation step to the E3. And that's been shown, or been suggested to be UBCH8, which is an E2 isoform. And the E3s have yet, actually there are 2 candidate E3s at present. Now, one of the obvious questions in this area is how do these different E1s, since they're very similar in sequence, distinguish their cognate ubiquitin-like protein from all the rest of these ubiquitin-like proteins? Well, to answer that question, we took the crystal structure for the activating enzyme for one of them, NET8, and using the structure for NET8, which is in green, bound to its active site, we modeled, or actually in red here, we modeled in the C-terminal domain of ISG15. And they fit pretty nicely because the two beta-grass folds coincide almost identically with a very low root mean squared deviation in residues for alpha carbons. And this allowed us then to define what the likely protein-protein interaction interface between ISG15 and its own activating enzyme would be, under the assumption that these protein-protein interactions are conserved across evolution. And the modeling also showed that the amino terminal domain, the one with the hydrophobic region, probably does not interact at all with the activating enzyme. And when we delete this, we find that the C-terminal domain itself binds to its activating enzyme with the same affinity as the wild-type protein, supporting that. Well, by mutagenesis, this is the face where it binds to the activating enzyme, and by mutagenesis, we find that the specificity filter is that single arginine at that position right there. If you mutate that to anything else, it doesn't bind. It not only doesn't bind, it doesn't work at all with the enzyme. All of the ubiquitin-like proteins have something other than arginine at that position and therefore are excluded from binding to this activating enzyme, so that this becomes the specificity filter for allowing this enzyme to distinguish ISG15 from the other ubiquitin-like paralogs. So moving back to this, the more interesting thing, which we found very recently, involves UBCH8. And actually what it does. If you look at all of the E2 families, and this was an informatics study that a student of mine, Tom Seatman, did some 10 years ago. At the time, he assembled all of the 125 extant E2 sequences known in the literature. And it took him about a month to assemble all these things and did an extremely large, at the time, sequence pileup. It's the first time anyone had applied informatics to the ubiquitin system. And keep in mind, this was 10 years ago. All of these E2 proteins have a 150 amino acid domain which folds into the same type of fold here. It's the amino terminus, helix, C-terminus. There's the active site cysteine, which forms a thioester to ubiquitin, which is transferred from the E1 to the E3 steps. And if you compare these sequences, they fall out into two large clades here, and each clade then falls out into distinct families, which from the literature have very specific phenotypes for deletion mutations. UBC2, for example, is involved in DNA repair, the end-end rule pathway, histone ubiquitination during chromatin remodeling. And within this pathway, which is particularly well represented, you see them falling out into animals, plants here, and yeast. The interesting thing about the analysis was there were 3 families that were not present in lower eukaryotes, meaning that this system has continued to diverge and evolve and acquire new functions with evolution. The one family down here, UBCH8, had UBCH7 and UBCH8, which at the time, 10 years ago, we thought were orthologs of one another. With increases in the number of sequences, we now know that these are actually 2 discrete families. The UBCH7 family is absolutely conserved among all of the orthologs known, mouse, human, bovine, chicken, so forth. The H8 family is not as well conserved, but is conserved at all of the important points to make it fold into the same type of structure. UBCH8 is the one that was proposed to be the ISG15 specific enzyme and its conjugation pathway, but it had originally been identified as a ubiquitin-specific conjugating enzyme. So the dilemma was, how can this enzyme work in both a ubiquitin and an ISG15 pathway? Now, what we reasoned was, because from this analysis, they're all very similar in structure and sequence. If you're not careful and don't apply careful enzymology and worry about concentrations, particularly if doing overexpression within a cell, you can drive otherwise unfavorable binding interactions and make these E2s that are not supposed to work with their particular E1 work with something that it shouldn't. And so to test this, a research associate in my lab, Jenny Klein, did really the first careful study, and she compared the ability of UBC2, that Rad6 ortholog at the top I showed you, which is a ubiquitin-specific conjugating enzyme, to UBCH8 and UBCH7. First with the ubiquitin activating enzyme and radiolabeled ubiquitin. And what this is a non-reducing gel, okay? Not boiled, no SDS, so that you preserve these thioesters formed between the E1 and the E2. And you can see in the presence of the activating enzyme, UBA1 for ubiquitin, you see a ubiquitin E1 thioester formed. And we know it's a thioester because if we boil these samples in the presence of SDS, all of these bands disappear, as one expects for a thioester. If one now adds 40 nanomolar of this ubiquitin-specific E2, you see not only the E1 thioester, but this thioester is transferred onto the E2 to form an E2 ubiquitin thioester here. And that goes away under reducing conditions. If you add the same concentration of UBCH7, you see formation of a UBCH7 thioester, but at the same concentration, you don't see formation of a UBCH8 thioester. And these are all 1-minute incubations because the turnover number for this is very fast. It's about 5 per second. So when others had done this, they'd found this reaction work. So we were wondering, how is it that other people find thioester formation between ubiquitin and this, but we're not under these conditions? It turns out if you run the concentration up tenfold to 400 nanomolar, you can just begin to see thioester formation to UBCH8. And if you do this not for 1 minute, but for 30 minutes at 10 times the concentration, which is how people did it in the literature, you see a very nice thioester formation. The problem is that this is under extreme conditions compared to how the real ubiquitin-specific enzymes work. So you can already see where I'm going with this. If we do the converse experiment of using UBE1L, the ISG15 specific enzyme, and radiolabeled ISG15, it forms a thioester to its activating enzyme. And that molecular weight difference recognizes the fact that ISG15 is 15 kilodaltons, whereas ubiquitin is just 8 kilodaltons. If you add 40 nanomolar of UBCH8, well, you see a thioester form there. We have this unfortunate contamination that occurs here because ISG15 dimerizes and forms a covalent adduct to itself over time. So this thing appears and we can never quite get rid of that band. But there's a thioester to UBCH8. These two exposures were done identically to one another to account for the difference in specific activity. So you can see that the amount of thioester form is qualitatively as well as quantitatively identical to what you have here for equivalent amounts of E2. And as a negative control, the ubiquitin-specific UBC2, you see no thioester. If you go to 10 times as much, you see no thioester. 10 times as much and 30 times longer, you just begin to see it. So you can drive these unfavorable reactions under very unphysiological conditions. And with UBCH7, which worked with ubiquitin over here, the same thing. No thioester at 40 nanomolar, 10 times more, not much. 10 times more and 30 times longer, you can see a thioester form. Again, you can drive this reaction. Bottom line, UBCH7 specific for ubiquitin, UBCH8 specific for ISG15. Now, interesting point. In the literature, there are a lot of instances of UBCH8 being identified as the E2 for ubiquitin targeting pathways. So we went into the literature and we started to look for and ask the question, what E3 ligases have been identified that work with both UBCH8 and UBCH7? One other thing, I can probably skip this. Instead of the qualitative thing we did before, we can actually measure the rate of thioester formation to an E2 under legitimate kinetic conditions, and you can watch with time this thioester form. And if you calculate the rates, you get very nice double reciprocal plots. This is for UBC2 charging by the ubiquitin activating enzyme. And when we do this, we can get, as we vary ATP and ubiquitin, we can get KMs of 7 micromolar for ATP, which means ubiquitin activation is always saturating with respect to ATP. The Km for ubiquitin is about 0.8 micromolar. Concentration of ubiquitin normally is about 40 micromolar in a cell, so it's always saturating with respect to ubiquitin. And for the E2, the Km is about 100 nanomolar, about 40-fold above the normal E2 concentration. Well, this is normally about 12 micromolar. So it's always saturating with respect to the E2. When we look at ISG15, we get normal saturation kinetics for ATP, ISG15, and UBCH8. And we can show that all of these activating enzymes that we've looked at, the one for ubiquitin, for ISG15, and for NET8, all have very similar affinities for ATP and their cognate ubiquitin-like protein. So that they are very similar. They only differ in specificity for their ubiquitin-like protein and their E2. And that the reason that the ubiquitin activating enzyme doesn't work with UBCH8 is that its Km is about 40 micromolar, whereas the Km for the normal E2s are around 100 nanomolar. This Kcat rather low. We find that the one residue, which is the specificity filter, is a methionine at position 4, which normally would be an arginine in ubiquitin-specific E2s. And if we mutate that to an arginine, we can make UBCH8 shift its specificity from ubiquitin to ISG15. The first time anyone's ever pulled off this little trick of synthetic evolution. The converse holds for UBCH7. You can mutate it to the methionine, and it becomes much more like UBCH8. And with UBE1L, there's quite a bit of difference between UBCH8 and 7. But for time's sake, let me move on. These are the E3s which have been shown in literature to work with both UBCH8 and UBCH7. And what we propose is that these proteins, which there's a very interesting group of them, function normally as ubiquitin-specific ligases for targeting their respective proteins for degradation, but under an interferon response or in response to retinoic acid, which also induces this ISG15 system, they shift specificity to UBCH8 and begin putting ISG15 onto those positions normally targeted by ubiquitin and stabilize the protein against ubiquitin-specific degradation. Some of these are really, really interesting. Dorphin is involved in superoxide dismutase degradation, and which is the thing involved in ALS. EFP is involved in estrogen-induced breast tumor transformation. The one we particularly like is E6AP, which is how human papillomavirus transforms a cell because it produces a protein called E6, which binds to E6AP, shifts the specificity of E6AP to now degrade P53, and in that way transforms the cell. Turns out that hepatitis virus C produces its own protein called HS1B, which does the same binding to E6AP, but shifts the specificity of E6AP to now target retinoblastoma protein and transform hepatic cells into being cancerous cells. And there's some other ones down here, Parkin, which is involved in Parkinson's disease, and a few others. Several of them are involved in vesicular trafficking. Okay, now, so we've concentrated on E6AP, and very quickly here, to ask the question, does E6AP target, does it work with ISG15? The architecture of E6AP is it has a 300 amino acid domain at the end, which is shown here, and work from Ivan Pavletich's lab, binds UBC87 normally, transfers a ubiquitin from that cysteine, presumably, onto an active site cysteine on the E3, and then it's transferred onto the target protein. We think this structure actually is incorrect, that it's a product complex, for reasons I can discuss later if you wish. Turns out, very quickly, E6AP forms a thioester with ubiquitin at that cysteine I showed you. E6AP equally forms a thioester with ISG15, but at a slower rate, and it does so because whereas this is E3 limiting here for E6AP, because the KCAT for UBE1L is very low, this is actually limited by the ability of the activating enzyme to activate ISG15. And if you correct for that, this actually is proceeding at the same rate that it is here. So the problem is not with E6AP forming a thioester with ISG15, but with the slow rate at which the activation occurs. E6AP is able to form polyubiquitin chains with UBC87, which are shown here, and it makes everything from a trimer of ubiquitin up to 10 or even so many ubiquitins in length that they won't move into this gel. This is a reducing gel now. With ISG15 pathway, E6AP is able to form polyubiquitin chains of ISG15 up to 12 or longer. And again, it's slower, not because of the E6AP, but because of the activation step. So how we think these bifunctional ligases work is normally they target for degradation via ubiquitin. In the presence of an interferon response, you begin to substitute ISG15 and UBCH8 in the pathway. You conjugate to the same lysines, and because ISG15 is not targeting for degradation normally, you stabilize that protein against degradation. Okay, so what I've done is to briefly introduce the idea of ubiquitin-like proteins, shown the pathway for ISG15 conjugation, shown that if one's not careful in the sorts of experimentation people do nowadays where you're not worried about concentrations in enzymology, you can come up with erroneous conclusions about what interacts with what. And if these are done right, you get an insight, that is, that a certain E2 will only work with this pathway, which we believe has introduced this idea of a bifunctional ligase that can work between two different ubiquitin-like proteins to have antagonistic roles, and that we believe that this explains, in part, how the interferon response works within cells. And I thank you for your attention.